Welcome everyone to episode 8. Last episode, I introduced the cell as the fundamental building block of all living things. I discussed the qualities that eukaryotes and prokaryotes share, and the organelles inside eukaryotic cells that allow them to perform such complex functions. In this episode, I'm going to dig deeper into cell physiology by exploring the internal subsystems and structures of the cell. In terms of cellular physiology, you can think of this episode kind of like a part 2 to episode 7's part 1. I'll make many references to material covered in episode 7, so if you haven't listened to that episode yet, I suggest you do so before listening to this one. In episode 7, I talked about the cytoskeleton that prokaryotes and eukaryotes share in common. In this episode, I'll talk more about the cytoskeleton, including the molecular structure of its fibers and the functions that they do for the cell. In the last episode, I talked about the endoplasmic reticulum and the roles that it plays and how it interacts with the Golgi complex. In this episode, I'll explore how these organelles interact and communicate on a molecular level, and how they organize and distribute specific proteins or lipids to the right places. In the last episode, I briefly discussed the nuclear envelope. In this episode, I'll discuss the nuclear pore complex that regulates the passage of particles into and out of the nucleus. As I discuss this physiology, try to imagine how these systems interact simultaneously to allow the cell to stay alive and remain functional. Perhaps the easiest way for a cell to stay alive and functional is to have a stable, open compartment in which all of its biochemistry can go along undisturbed. The phospholipid bilayer membrane does a good job at regulating the passage of particles into and out of the cell, but it lacks a certain consistency. The phospholipids in the bilayer, they're not actually connected to one another, and they float just kind of side by side in response to hydrophobic interactions with the water. They kind of shuffle along together if the cell moves slowly, but they can't really move in a coordinated, deliberate way. More importantly, they can't really move the cell in a coordinated, deliberate way. The cytoskeleton, however, can do all of this. So what is a cytoskeleton? In episode 7, I briefly touched on the cytoskeleton, describing it as a series of thin filaments layered across the inside of the membrane. This is true, but the cytoskeleton does more. The fibers crisscross the cytoplasm in a flexible network that stabilizes the cell and organizes the mechanical relationship between organelles. As a whole, the cytoskeleton provides the consistency that the fluid bilayer doesn't have. In concert with the phospholipid bilayer, the cytoskeleton allows the cell to exhibit a fantastic plasticity. It can smoothly expand or contract in response to osmosis or in response to the density of nearby cells. It allows the cell to move and reorganize internal structures, which is especially important during cellular replication when all the internal structures are getting copied and divided between the daughter cells. Together, the fibers of the cytoskeleton create a flexible meshwork that weaves throughout the insides of the cell, stabilizing it and enabling a dynamic integration between the cell's internal components. The cytoskeleton is composed of 3 distinct types of fibers. Each fiber has a unique structural design that imparts qualities unto it. When numerous fibers layer together, or mix with other fiber types, they can create larger structures like sheets or webbing, and this imparts additional qualities into the cell. So from largest to smallest, these fibers are microtubules, intermediate filaments, and actin filaments. Microtubules are large, coiling polymers composed of dimer monomers. That is, the microtubule polymer has subunits which are dimers, molecules composed of 2 separate proteins bound together. In the case of microtubules, the monomer dimers are composed of 2 proteins called alpha and beta tubulin. These dimers are polar molecules, possessing both a negative and positive end. When they align in their tens and hundreds to form a polymer, they organize according to their polarity. Because opposites attract, each monomer aligns itself in the same repeating orientation relative to the others, with the positive and negative ends all pointing in the same direction along the chain. One end of the polymer is therefore going to hold a negative polarity, while the other end holds a positive polarity, as these are the exposed ends. The positive end has a free beta tubulin, while the negative end has a free alpha tubulin. With this information, you can deduce that the alpha tubulin protein has a high concentration of amino acids, with negatively charged or negative polar side chains. Similarly, you can deduce that the beta tubulin protein has a high concentration of amino acids with positively charged or positive polar side chains. Due to electronegative interactions with ATP, the positive end of the microtubule grows faster than the negative end. In order for the microtubule polymer to grow, a molecule of ATP must bind to a free monomer. This complex then binds to the end of the polymer, using the energy in the ATP to form the bond. When the polymer is degraded, this ATP is consumed in a hydrolysis reaction to form ADP and an inorganic phosphate ion. So in all species, microtubules originate from a particular molecule or group of molecules known generally as a microtubule organizing center, or an MTOC. The MTOC holds the negative ends of the microtubule, allowing the positive ends to grow outwards into the depths of the cell. Each MTOC is swamped in a cloud of proteins involved in the synthesis of microtubule fibers. Plants can have hundreds of these MTOCs throughout their cells, but animal and fungal cells typically have just one microtubule organizing center per cell. In the animal cell, the MTOC is called the centrosome, as it is the center of the radiating microtubule fibers that reach out into the cell. The centrosome assists in cellular replication by reaching out with microtubules to the numerous pairs of replicated chromosomes. These microtubules anchor to the chromosomes and they're attracted by the centrosomes, which pull the chromosomes apart towards opposite ends of the cell. This is a critical step in cellular division. If one daughter cell doesn't get its DNA, it can't really grow or reproduce or do anything besides briefly take up resources and die. The animal centrosome is composed of two identical structures, oriented at 90 degrees to one another. Each structure is a centriole, a tube composed of 9 tubulin trimers arranged with their long axis parallel to one another, and their short axes radiating outward from the ring. These centrioles were once thought to be required for microtubule synthesis, although recent experiments have shown that this may not be the case. At any rate, the centrioles are involved in organizing the radial growth of the microtubules. These microtubules are critically important for the transport of cargo-laden vesicles throughout the cell. Kinesin molecules use the microtubules like highways or roads to drag vesicles where they have to go. Kinesin is a motor protein, a special class of proteins that can consume ATP to induce a forceful movement. The kinesin molecule has a head, neck, and tail region. The tail region can anchor to a membrane, like a vesicle. The neck connects the tail to the head, which has two globular structures that are kind of like feet. By consuming a molecule of ADP through a hydrolysis reaction, the kinesis molecule can undergo a conformational change in the head region, effectively taking a single step. A step along the microtubule fiber. So it eats another ATP, and it takes another step. Another ATP, another step. The feet of the kinesin molecule bind to the microtubule, and each ATP hydrolysis reaction makes the kinesis take one more step down the microtubule road, heading from the negative end of the microtubule to the positive end. Another motor protein called dynin can walk along the microtubules in the opposite direction, from the positive end to the negative end. However, I'll discuss the dynin protein in more detail in a few minutes. Binding to a molecule of ATP causes the first conformational change in kinesin, initiating the step. The release of ADP as a product of the hydrolysis reaction creates another conformational change, which completes the step. In living cells, the kinesin molecule can take up to 375 steps per second, which is really fast. Due to their function as roads used by the vesicle-dragging kinesin proteins, the microtubules are incredibly important for the proper functioning of the endoplasmic reticulum and the Golgi complex. The rough endoplasmic reticulum is responsible for synthesizing proteins, while the smooth endoplasmic reticulum processes lipids. Together, these two regions of the ER send their products to the Golgi apparatus to be sorted and labeled for transport. Microtubules are used as the road tracks for the transport of these vesicles. At the Golgi apparatus, proteins and lipids are organized according to where they need to go in the cell, and they're repackaged into another vesicle and sent on their way. The Golgi apparatus uses microtubules to send its vesicles to the right destinations. Two important cellular features that I cannot skip are the cilia and the flagella. Both are a type of whip-like structures composed of microtubules that can induce movement by rapidly wiggling and waving back and forth. Flagella are generally large ropes of microtubules extending out of the side of the cell. In prokaryotic cells, the flagellum is like a helix or a screw, and when it rotates, it propels the cell along like the rotor on a boat. In eukaryotic cells, the flagellum waves back and forth, waving like the tail of a fish to beat its way through the water. Consider the human sperm cell. It's a haploid germ cell with a characteristic flagellum that it waves back and forth to surge forward in powerful bursts of movement. Where the flagella are large and powerful, the cilium is small and weak. This difference in size is made up for in number and distribution, where one cell can have usually one or maybe up to 4 flagella. Another cell can be covered in tens of thousands of cilia. The cilia are like little hairs, and they're able to wave and pulse in rhythmic coordination with other cilia. Through this, the cilia can induce movement by fanning themselves against a fluid solution. Cilia on the outside of the cell can induce movement by propelling the cell through the water. Cilia on the inside of the cell can induce directed currents in the cytoplasm, kind of like the collective waving of the myosin proteins on an actin filament. Both cilia and flagella have a similar tube-like structure, and they use a protein called dynin to facilitate their waving and wiggling. I mentioned dynin earlier, if you might recall. So the stem, or the stalk of a cilium or flagellum strand, it's called an axoneme. So if you were to cut the axoneme in half and look at its cross-section, you would see a ring surrounding 2 points, with what looks like spokes connecting them. So this ring is composed of 9 microtubule pairs, arranged like the centriole. The long axis of each microtubule pair is parallel, running the length of the axoneme, but the short axis of each microtubule radiates at an angle away from the center of the microtubule. In each microtubule pair, one microtubule is a complete tube. The other microtubule is incomplete, like a paper roll, with a lengthwise strip removed. This incomplete microtubule sits alongside the complete microtubule, and so both of them look in the cross-section like 2 overlapping circles of a Venn diagram. So this is each pair, and there's 9 pairs in a ring. The 2 points in the center of the ring are also microtubules. The 2 points in the center of the ring are also microtubules, each paired with another microtubule like those on the outer ring. Filaments connect the ring microtubules to the central microtubules like spokes on a bike, giving the axoneme a certain stiffness and stability. Additional bonds and links between the microtubule pairs in the ring reinforce this stability. So how does the cilia wiggle rhythmically to the pulse of the cell? How does the flagella wave around to push the cell around? This action is the result of a protein called dynin. Because of all the bonds and linkages stabilizing the microtubules, they cannot be easily pushed or pulled around by protein motors. The effective range of movement is heavily constrained, as the movement of one microtubule pair will tug on and move all of the nearby pairs. So the dynin motor protein, which is one of the types of molecular links connecting microtubule pairs, by the way, will undergo a conformational change and apply force to the microtubules. Because of structural constraints, the microtubules can't just slide alongside one another, or pull themselves close together. Instead, the dynin contorts the microtubules, causing them to bend. When the dynin proteins on each side of the flagella are alternatively activated, it produces a back and forth waving motion. One side is activated, and the proteins flex, and the microtubule bends one way. The other side is activated, and so those proteins flex, and the microtubule bends the other way. Do this quickly, over and over again, and you basically have a long waving paddle that's giving you enough momentum to scoot around the mud puddle that you call home. The second type of cytoskeletal fiber is called the intermediate filament. Intermediate filaments are nonpolar molecules used exclusively for various structural purposes within the cell. Intermediate filaments come in a large variety of different types, depending both on the species and the type of cell. These polymers can have many different kinds of monomers, specialized for whichever purpose that particular cell has. Your skin cells, for example, have 20 different types of keratin filaments in them. Your fingernails, toenails, and your hair are all different types of keratin being secreted by your cells. Within your cells, intermediate filaments provide structural support and protection against mechanical damage like scrapes or strikes. Intermediate filaments line the membrane of the cell, giving it a degree of stability and rigidity. A type of intermediate filament called a nuclear lamin does the same thing for the nuclear envelope, giving structural support to the double membrane of the nucleus. The nuclear lamins help the nucleus keep its shape by forming dense sheets, just like keratin and carbohydrates like cellulose and chitin. The nuclear lamins are involved in the dissolution and reconstruction of the nuclear envelope during cellular replication, presumably working to hold on to a few phospholipids to act as sites of membrane regeneration. These filaments also anchor the chromosome molecules to various points on the inside of the nuclear envelope. This helps keep the chromosomes from getting disordered and tangled up, which would impair cellular replication and proper DNA expression. Intermediate filaments also extend from the nucleus to connect to the filaments forming an inner layer on the membrane. These extending filaments anchor the nucleus in place within the cell and provide another degree of internal stability and organization. The third types of fibers are actin filaments, also called microfilaments. They're very thin, typically 6 nanometer wide dual polymer molecules composed of a protein called actin. So actin forms subunits called G-actin, and here the G stands for globular, so they're like little globs of proteins, little globs of actin proteins called G-actin. These subunits assemble into longer filaments of actin, and they're called filamentous actin, or like filament actin, or F-actin for short. So the creation of an actin filament, or F-actin, begins when 3 G-actin subunits spontaneously form a 3-monomer chain called a trimer. Trimers connect together to create the longer F-actin filaments, which compile together into bundles or networks. A suite of proteins is involved in organizing and binding the fibers together. Actin possesses a polarity, with each molecule having a positively charged end and a negatively charged end, kind of like the microtubules I talked about earlier. In groups, actin molecules organize themselves into a line, with a charged end of one molecule forming a non-covalent bond with the oppositely charged end of another molecule. In this way, actin proteins form a polymer of repeating monomers with a shared orientation. 2 strings of actin coil around one another to produce a single actin filament, each string giving the filament a positively charged end and a negatively charged end, also called a barbed end and a pointed end, respectively. These non-covalent bonds make the actin filament a particularly unstable molecule. This instability is integral to the functional qualities of actin filaments. You see, each filament grows or shrinks depending on the concentration of G-actin subunits available. Much like microtubules, the positive end of the actin filament, or the barbed end, is able to grow much faster than the negative, or the pointed end. The barbed ends grow when it binds to a G-actin molecule bound to ATP. The ATP molecule is subsequently cleaved off in a hydrolysis reaction, where it returns to the cytoplasm to be used in other reactions. And so this happens pretty quickly, whereas the polymer degradation at the negative end happens relatively slowly. G-actin on the negative end are slowly broken off and released into the cytoplasm. So now we have both ATP and G-actin monomers floating around in the cytoplasm. They can associate and bind to the positive end of the polymer. In this way, actin filaments have something called dynamic instability. A specific protein called myosin is associated with actin. The myosin protein is really interesting, and it serves a really important purpose. Like kinesin, myosin is a motor protein that consumes ATP to produce a force or a specific movement. Myosin proteins have 3 distinct regions called the head, neck, and tail. The head region consumes an ATP molecule in a hydrolysis reaction, creating ADP and an inorganic phosphate ion. When the head region detaches from the ADP, its conformation rapidly changes. The head and neck regions of the myosin protein are involved in a rapid and forceful conformational change, like a leg kicking out forcefully. The myosin head can bind to an actin filament. Each kick from the myosin head pulls the actin filament a little bit in one direction. Your muscle cells are packed with millions of myosin and actin molecules, aligned in parallel along the direction of your muscle tissue. So when you contract your muscle, these myosin molecules fire off in their millions, pulling the actin filaments with millions of little tiny tugs. This collective effort all across your muscle is what causes the contraction. It's what allows you to lift stuff, to move stuff. If you'll humor me with a silly analogy, imagine 2 space stations floating out in zero gravity. Each space station has a long metal cord extending out some distance towards one another, with the end section of the cords overlapping a little bit. Along one cord are tied a large number of astronauts, and so when they get the signal, all the astronauts who can reach the other cord will grab it and pull it down as hard as they can. This collective action will tug the space stations a little bit closer together, give the astronauts a moment to catch their breath and get a fresh grip, and then give the cord another huge pull, tugging the stations even closer together. The astronauts are like myosin proteins, pulling on the actin filament. The 2 space stations are like your bones, being pulled together by the muscle. Except muscle cells have literally thousands of these actin-myosin complexes, and muscles have millions of cells. Together, the billions of actin-myosin complexes in any given muscle have enough collective power to allow you to do stuff, like walking and running, blinking and speaking, lifting things up and putting them down, throwing your stuff, or delicately manipulating it with fine motor control, that kind of stuff. If you want to imagine your bicep with the astronaut analogy, you'd have to imagine the space stations are connected by thousands of pairs of fibers, each dotted with hundreds of astronauts, all collectively pulling to bring the space stations closer together. In all cells, actin filaments use the myosin proteins for motility. During cytokinesis or cell division, many actin filaments wrap around the cell like a belt. When cell division is ready, the myosin allows the actin filaments to slide past one another, tightening like a belt or a rubber band around the middle of a water balloon. The tightening ring of actin filaments eventually pinches the cell membrane in two, producing two independent daughter cells. Actin is responsible for a different kind of cell motility. Groups of actin filaments can grow and bulge outwards, pushing out a lump in the side of the cell. These lumps can gently push the cell around, kind of like feet. A series of directed pushes by these actin lumps, or these weird little feet protrusions, can make the cell crawl around, and it gives it a degree of independent motility. Recall how I said that fibers in the cytoskeleton also move across the cell's internal space, through the cytoplasm, and connecting opposite sides of the cell. When actin filaments and myosin proteins crisscross the cell, they can influence the currents and flow patterns of the cytoplasm. Each kick of the myosin is like a canoe paddle. The flow of cytoplasm is directed along the length of the actin filament by the collective paddling of all the myosin. In this way, the specific orientation and placement of actin filaments can assist the directed transport of vesicles distributed by the Golgi complex. If the Golgi complex can send a vesicle of proteins along a known current in the cytoplasm to its target, that can help optimize the cell's functional effectiveness. So that's about it for the cytoskeleton. The next substructure within the cell that I want to discuss is the endomembrane system. The endomembrane system, in a nutshell, is composed of the endoplasmic reticulum, the Golgi apparatus, all the transport vesicles they produce, and all the microtubules they use to shuttle stuff around. I've discussed microtubules already, so I'll focus on the production and processing of vesicles and their cargo. Every part of the cell needs something different. Acid hydrolases and other proteins must go to the lysosomes, where they sustain an internal pH around 4.5. Membrane proteins and phospholipids have to go and be installed into the cell's bilayer membrane. Catalases and other proteins must go to the peroxisomes, where they break down long-chain fatty acids and reduce oxidizing radicals. The Golgi apparatus packages and sends all the necessary parts to their proper location. But how does it do it? For that matter, how does the endoplasmic reticulum shuttle proteins to the Golgi apparatus? It turns out that when proteins are synthesized by ribosomes on the outside of the rough endoplasmic reticulum, they're given a chemical tag that directs them to enter the endoplasmic reticulum itself. This chemical tag binds to a small molecule called an SRP, or a signal recognition particle, locking in place and halting further synthesis of the polypeptide chain. The SRP ribosome complex comes near the membrane, eventually coming into contact with an SRP receptor. When the SRP binds to the SRC receptor, the SRP breaks free from the complex and floats away. The polypeptide chain continues to be synthesized, and the protein is squirted into the lumen of the endoplasmic reticulum. So the SRP is kind of like a pause button for the ribosome. It halts production of the protein until it can find an SRP receptor to hold onto the ribosome for it. After it hands off the ribosome, the SRP detaches and floats off to bind to another ribosome. Inside the rough ER, in the lumen, these fresh polypeptide chains are folded properly into functional final shapes, with the assistance of a suite of chaperone proteins. Polypeptide chains or proteins entering the lumen of the rough ER come into association with numerous carbohydrates. When enzymes facilitate the binding of proteins and carbohydrates, they produce mixed glycoprotein molecules. The rough ER possibly uses these carbohydrates as tags, and the tags are used to properly associate and identify groups of proteins and package them together into vesicles. These vesicles are sent to the cis side of the Golgi complex, where specific surface proteins on both the Golgi and vesicle membranes allow them to link together and merge. The vesicle deposits its contents in the cisterna, and through the process of cisternal maturation is carried through the Golgi apparatus to the trans side. As the cisterna goes through stages of maturation and moves from cis to transface, it exhibits different groups of enzymes. This continuous series of enzymes modify the carbohydrate tags on the proteins, like a vehicle assembly line carving up some piece of metal. At the end of their enzymatic processing, the cisterna reaches maturation at the transface of the Golgi apparatus. At the transface, the cisterna begins to break up into smaller globular vesicles. The proteins are sorted according to specific chemical tags they're given, each tag corresponding to a specific location within the cell. The proteins are sorted into regions of the transface and grouped into vesicles according to their chemical tags. A mannose sugar with a phosphate group bound to its sixth carbon acts as the chemical tag directing the protein to a lysosome. 2 amino acid sequences, 9 and 3 bases long, are chemical tags directing the proteins to a peroxisome. Other specific tags are used to direct proteins to the cell's plasma membrane, or to regions within the cytoplasm. Each tag is recognized by organelle-specific receptor pores, which allow the tagged protein to enter the organelle. The final substructure that I want to discuss is the nuclear pore complex, which connects the inside of the nuclear envelope with the rest of the cell. As I've said before, the nuclear envelope holds the cell's genetic material, or the DNA of the organism. The nuclear envelope protects this DNA by keeping it in the heart of the cell, double wrapped with 2 lipid bilayers. The DNA inside the nucleus is opened and transcribed by a large and complex suite of proteins. The DNA is used to produce RNA molecules, like the RNA used in the large and small subunits of ribosomes. Messenger RNA, commonly referred to as mRNA, is a segment of the DNA that has been read and translated into a small strip of RNA by the enzyme RNA polymerase. This mRNA is the precursor to a protein, but first it has to leave the nucleus. The ribosomal subunits must also leave the nucleus, along with the mRNA. Together, these large molecules pass through large structures called nuclear pores, which penetrate both layers of the nuclear envelope. These pores also regulate what comes into and out of the nucleus. Molecules produced outside the nucleus, like proteins required for transcription and DNA repair, or the nucleotide bases required for making more DNA and RNA, are selectively allowed through the pores to the inside of the nucleus. This raises a question. How do these pores know to let in these molecules, and not others of similar size and polarity? The details are being researched as we speak, but what is known is that proteins and other molecules produced outside the nucleus cannot enter the nucleus unless they have a particular chemical tag. This tag takes the form of a polypeptide chain composed of 17 amino acid monomers. All proteins bound for the nucleus, like the ones required for transcription and DNA repair, are given this chemical tag. As a result, the amino acid sequence composing this tag is called the nuclear localization signal, or the NLS. The polypeptide chain is literally bound to the end of the finished protein, getting recognized by other molecules within the cell, who then direct the protein to the nucleus. The nuclear pores recognize the NLS and undergo a conformation change to allow the tagged protein to come through. Similarly, an NES, or a nuclear export signal, is used to facilitate the transport of mRNA out of the nucleus. Unfortunately, not much is known about the inner mechanisms of the nuclear pores, but research is ongoing and things are being learned every day. I think that's all about there is to the basic internal physiology of the cell. Considering the organelles and cell walls that I covered in the last episode, and the topics that I covered in this episode, I think I've decently covered the basics of all the physical structures in the cell. Next episode, I'll be discussing the chemical pathways for cellular metabolism. If you enjoyed this episode, hit the like button and subscribe. Share the episode with a teacher, a friend, or a classmate. If you want to support the show, follow us on social media and consider buying something from the official store. Follow us on Twitter. Our username is @BioLogic Podcast. To buy something from the store, go to redbubble.com and search for BioLogic. Or go directly to the store at redbubble.com/people/biologic/shop. Become an official patron of the show. Sign up at patreon.com/biologicpodcast. Your support really helps the show, so if you like it, send us some love. And as always, thanks for listening.